So one of the things we need to keep in mind whenever we're in evolutionary biology is that evolutionary biologists are making observations of things in real time, right now. And we're trying to explain what we're seeing, this diversity of life, the unusual behaviors, the unusual adaptations and structures. We're trying to explain how we got here. This underlying assumption has to be explicitly acknowledged. We have to keep returning to that when we're looking at the mechanism of evolution, genetic drift. The reason why is because genetic drift is a change in gene frequency. In other words, we would anticipate that leading up to the moment when some random event occurred, we had a tremendous amount of genetic variation. But that random event shifted the frequency in genes. And so maybe some genes disappeared from the population, some variations, some adaptations were gone. And from that moment going forward, all we observe is the same, or is all we see are the same genes. So we see that shift in the frequency in genes in the population. So genetic drift is that change in gene frequency in a population. And the key aspect here is the randomness. And I love the fact that we've got a part of evolutionary biology that openly embraces the idea that life just sucks sometimes. You know, I've taken statistics, and in statistics you learn about probabilities. And then maybe you start to learn about things like the idea that for safety, especially, say, the safety in our car, they've calculated the probabilities of things like what's the probability your brakes will fail? What's the probability your engine will just shut off and stop working? What's the probability that all of these mechanical components will break? And that's driving very nerve-wracking, because while the probability that my brakes may not work could be 0.0000001% of the time, it's not zero. It still could happen. What if today? And what if it's me? What if I'm the unlucky one that's on the wrong side of that probability curve, and today is the day that the brakes fail? Well, that sucks, right? Like, it's a low probability of happening, but it's not impossible. It's still in the realm of possibility. And so when we take that concept of probability, what's the probability that these deer or this puma is going to be at the base of the avalanche when the snow breaks loose and everything gets destroyed? What's the probability that these animals or these plants are going to be on the wrong side of Mount St. Helens on the day that Mount St. Helens goes kablooey and half the mountain gets blown away? What's the probability that these animals, these plants are stuck in the landslide when the wet ground gives way, or the flood exceeds the banks and takes over a place that you never imagined? Or what's the probability that you're stuck in Texas and all of a sudden the temperature drops and you got tons of snow dumped on you in Texas where you never imagined it would happen? Or what's the probability that you're underneath one of the hurricanes that's dropping feet, not inches, feet of rain within just a couple of days? Like, crap happens on this planet, and it just sucks if you happen to be in the wrong place at the wrong time. That probability game, somebody's going to get hurt. And genetic drift, our concept, our mechanism, that a role of randomness. Now, if we have a larger population, a lot more individuals, then when this random event occurs, eliminating some of those individuals, genetic drift is less likely. Part of that is because of the heterozygous genotype. The probability that rare or recessive genes would still exist in a population even after the crappy event happens is much more likely. And if we have a small population and that random event occurs, the idea that those rare or unusual or infrequent genes disappear is much higher. A lot of times what we see after the random event is we see a homogeneous gene pool. We see a decrease in variation. A decrease in variation could lead to detrimental, deleterious alleles. And this is why I really liked that research that we did in the first half of the semester, where we looked at dogs. And the researchers were asking the question, what happened? What led to that increase in deleterious alleles? Was it a type of genetic drift, or was it just inbreeding through artificial selection? So when we're dealing with genetic drift, we have two main types. The first type is something called a bottleneck. And with a bottleneck, what we see is a shrinking in the genes in the population. That's due to some kind of random event. Sometimes that's due, like in the case of the Isle Royale wolves, sometimes that's due to disease, and sometimes that's due to the human habitat destruction, as we see with the prairie chickens. Regardless of what the random event is, a lot of individuals die, and so what we see going forward from that random event is a similarity in genes in all the individuals in that population. Now, the other type of genetic drift that we see, that we tend to see less frequently, is something called a founder effect. And the founder effect is when we have two or more individuals that go to a new location. When they go to that new location where this species has never been before, they start to mate, they start to raise the population, increase the amount of individuals in the population. But what we see going forward from that founder effect is that all of the kids and the grandkids and the great-grandkids are very genetically similar and very closely related to the original two. And so within that population, we have a tremendous amount of inbreeding. How inbreeding can lead to an increase in deleterious alleles. So both the bottleneck effect and the founder effect have the same result, which is a decrease in genetic variance. And there is one thing that we know fairly concisely, that we're fairly sure of, which is as soon as you decrease genetic variation, you decrease the flexibility of the species to survive and adapt to changes in the environment. And so through the lecture slides, I give you lots of different examples of what that decrease in genetic variation leads to. Now, from an outside point of view, from humans looking in on the system, it's very to look at that and go, well, we need to get more genetic variation into this population. This is why I love the Isle Royale and the wolves, because the wolves coming to the island in the middle of Lake Superior, Isle Royale, those first two individuals in the 40s demonstrated the founder effect. And then the canine parvovirus in the 1980s demonstrates our bottleneck effect. So we have both types of genetic drift in this population of wolves. We have a tremendous amount of genetic similarity. And what we saw happen was the increase in vertebrae. And then within the last couple of years, the population decreased to just the two wolves, who were so closely related that even though the male wanted to mate with the female, she would not accept him. And the hypothesis for that is because they were so genetically similar. And then the male did die, and so there's just one remaining individual from the original population. And at the time, the researchers were really struggling with, what do we do here? Do we just let the wolves go extinct on this island? If we do, the moose population is going to explode and go through some problem. They'll start to starve, and then there'll be a lot of disease, so then they'll decrease. And then what's that going to do to the plants and all the rest of the other animals and organisms on this island? It ended up becoming a really challenging problem. And they decided to go ahead and grab some wolves from the mainland in Canada and over in Minnesota and Wisconsin, grab some individuals and do a wolf reintroduction. Now this is beautiful. We don't often get an island system clearly bounded with no flow back to the mainland. We get to observe what has happened leading up to the reintroduction. Excuse me. And then with the reintroduction, we get to see the results. They've got the individuals that they reintroduced tagged. They're tracking their movements. It doesn't look like anybody is breeding yet, but we do have some pack formation starting to come together, some beginnings of pack formation. So what we're hoping will happen over time is that we will have that genetic variation that will allow this population to remain healthy, which then affects the moose and affects the rest of the island over time. So please make sure when you are looking through those examples that you know the details, you know the nuance, you're making that distinction between a bottleneck and a founder effect. You understand how they can both have the same result of decreasing genetic variation, and that from an outside point of view, we may want to intervene and introduce genetic variation. When we are observing organisms right now, part of what we may be observing or seeing is the result of that similar genetic variation. Which then brings me to the concept of bringing organisms back from extinction. Yes, we have been able to do that. The Pyrenean ibex went extinct in 2000, and researchers were able to bring one individual back, but unfortunately only lived for 4 minutes. There was some misconnection between the lungs and other organs in the body. Scientists are still pursuing whether they can take recently extinct organisms and bring them back to life. But if all you have is just one individual or 2 individuals, how are you going to get genetic variation? How are you not going to end up with this homogeneous population that has no genetic variation? And that is a very big challenge. In other words, when we're bringing organisms back from extinction, we're almost doing a reverse genetic drift here. We are the random event that then is bringing this organism back with almost no genetic variation and potentially a lot of inbreeding. So it's a very interesting part of science that's been developing over time, and our concepts of evolution are informing how and what we do and whether or not this is even going to be possible. As always, if you have any questions, please email me.